Go for it.
Okay, thank you. Well, we've got David Buchwalter is giving the presentation today, and it's great that we can have David coming all the way from North Carolina, and hopefully the storm, the cyclone or hurricane, as you call it, there is not going to disrupt the power or the internet or anything like that, because there's quite a storm happening there. I wanted to start saying that I first became aware of David's work when I was doing my early years of my PhD, and I was kind of getting involved in ecotoxicology, and somebody who was very discouraging of ecotoxicology, basically, to paraphrase them, said that all ecotoxicologists are crap except for somebody by the name of David Buchwalter. And so I looked up his work and was quite impressed, and have been very impressed since then. He does some really great stuff, and particularly with these streaming vertebrates that you can see on the screen, which are widely, as many of us at University of Canberra use or involved with in terms of biomonitoring and all sorts of things like that. But it's mainly the working with them in the field. And what David does, which is really cool, is looks at the physiology, the toxicology, the gene expression in these organisms, which are normally studied in the field, and relates that to metal pollution, to salt, salinization, some work with pesticides, and more recently he's been doing a lot of work with temperature tolerance, and that's what he's going to be talking about today. And he really brings together quite a few different themes which are in the IAE, from the genetics and the gene expression to the freshwater ecology to the ecochemistry and ecotoxicology side of things, brings them together in one person. Those of you who have been around the IAE for a few years will may remember meeting David when he was in Canberra, I think 4 or 5 years ago, but some time ago now. When was that, David?
It was 2015.
2015, so 5 years ago now, wow. So I won't say anything more, and I'll hand over to David to a talk that I'm really looking forward to. Thank you.
So I was trying to find the talk that I gave back in 2015 because I think at that time we had generated some of the preliminary data that we used to get this grant, which is largely what I'm going to be talking about. But I couldn't find the file, so I don't remember what I talked about. All right, before I do get into this though, I definitely want to acknowledge the work of a former postdoc in my lab, Hassan Tina Chu. She really led the effort on this project and did all of the heavy lifting. So I'm just the mouth talking. What I'm going to try to do here is pull together a variety of different studies that we did, where we try to specifically link physiological processes to life history outcomes from full life cycle rearing experiments. Sometimes we have the data from the rearing experiments, and so we know the relevance of the temperatures, and sometimes we're actually pulling animals out of those experiments and doing work with those samples. And this work is very much a collaboration between us at NC State and David Funk and Bern Sweeney and John Jackson at the Stroud Water Research Center. So I don't have to, and just if anyone has any questions, this can be super informal. You can stop me and interrupt me and ask at any time. So don't feel shy about stopping if you want any clarification. Hopefully I can provide that. I don't think I have to tell an Australian audience that we have issues with water and we have issues with temperature. We basically are already failing to meet the world's demands for decent fresh water. And you guys had your issues with fires, last year. We have things burning right now on the West Coast. And these are issues that are not going to go away anytime soon. If we think about freshwater ecosystems, they are disproportionately important. They make up less than 1% of the Earth's surface in terms of their footprint, but they support about 10% of the Earth's biodiversity. And those who work in freshwaters understand that insects are really crucial to the way these ecosystems function. They perform this alchemy of turning detritus and fallen leaves and algae into fish and bird tissue. They do all this work in terms of nutrient processing, and because of that, they're widely used in biomonitoring programs all over the world. And when I started working with aquatic insects, I was sort of struck by our just fundamental lack of understanding of how these organisms work at a physiological level, and how can we better use them as indicators by understanding a little bit more about the physiological processes that underpin their performance under different kinds of environmental stressor regimes. We also know that temperature really dominates their biology. Temperature determines where these species can live, when in the course of the year they are active and developing. And so there's a lot to learn about this. So if we look at a sort of a classic textbook example of traditional views of thermal performance curves, you know, they seem reasonable. You have this range of temperatures between these Tmin and Tmax. Some people would call that a CT max. Some optimal temperature where the performance is the greatest. And when I look at the shape of that curve, it reminds me a lot of the shape of the curve for things that are essential to life, but that can also be toxic at high concentrations. I think of a lot of metals, you know, like copper and zinc, for example, or selenium. And there are issues with being deficient, not having enough of it, but there's a certain amount of it that you need. And then when that's exceeded, you have some toxicity. But coming at this from a toxicological perspective, you know, this image here is a little bit too simplistic, because we have to think about the exposure scenario. Are we thinking about acute short-term thermal challenges? Are we thinking about long-term, sort of more chronic seasonal related sort of temperatures? Are we thinking about intermittent exposures? And all of these things are certainly relevant. So I think we have to sort of take time to focus on these individual things. And I would say that how we study temperature dictates what we learn. And so if we look at a typical sort of North American, I'll give you temperature in Fahrenheit, sorry about that. But we have basically, you know, the lower line represents daily average lows, the upper line represents daily average highs, and you can see that you've got that diel thermal fluctuation. You've got that on a sliding scale as the season develops and warms and then cools down. And rarely do we actually study temperature this comprehensively. Usually it's sort of a short-term pulse, you know, wiggling within the lower to the high daily high sort of regimes, and a sort of acute push, or you know, statically across a long time. Insects respond to this sort of seasonal or calendar year sort of temperature regime, often by diapausing, sort of avoiding the unfavorably cold or unfavorably hot times of the year by being quiescent, usually in the egg stage, and then maybe taking advantage of things in terms of volcanism, having multiple generations in a year in some cases for some species. Aquatic entomologists have traditionally, as Ben said, done all of their work in the field, and there haven't really been very many good physiological model organisms for us to work with. I've worked really hard with the folks from the Stroud Water Research Center to try to develop this animal called Neocloeon triangulifer into something of a lab model. And it has certain characteristics that make it amenable for lab culture. It is an obligate parthenogen, which means there's no males and there's no issues about cues for mating or any of that. The eggs actually don't quite diapause, which is nice. So there's no diapause to break. And mayflies are a particularly good model because they develop in the water as larvae and they emerge from the water as winged subadults with their full complement of eggs. They no longer feed in the adult stage, and the adult stage is very short. So basically what pops out of the water is like a measure of fitness. It's not like a dragonfly that, you know, will feed for a long time as a terrestrial adult. The Stroud folks learned how to grow these animals on these periphyton plates that are grown on natural stream water. They're full of diatoms, which is the diet of this animal. And they've been rearing this animal for a long time. And were kind enough to share it with me almost probably 10 years or so. And what prompted me to get my hands on this animal was that the university that I work at, North Carolina State University, had just purchased its first high throughput sequencer. This is back when the 454 was like the big new shiny object. And the person who ran that instrument is somebody who I occasionally drank beer with. And he said, hey, if you want me to sequence something, it'll just cost you the reagent costs. And so all of the work that I had done prior to this had been on field collected animals that I had taken from nature and brought back into the laboratory. And so this was an opportunity to develop something that I could work with more extensively in the lab. And so we started sequencing some DNA and started to do a little bit, move into sort of more modern molecular methods. So getting back to what I was talking about in terms of exposure regime, this is the most classic sort of way that people think about temperature stress, is that, and we call these experiments death ramps, where we basically put the animals in water and warm them up. And you know, here's an example of a death ramp experiment in Neocloeon triangulifer. We've raised the temperature 1 degree C per hour, and we get a CT max estimate pretty close to 40 degrees C, a temperature which we never ever see here. All right, it's just, we don't get water that hot. In contrast, if we rear these animals across this temperature gradient from egg hatch all the way to adulthood, which is what I'm showing you in panel A here, we see a totally different scenario. Each one of these dots represents the mean of 5 to 9 rearing jars, with each jar having 50 larvae in it. So this is a really big experiment with thousands of animals. And what's interesting about this is that survivorship holds on pretty strong until just past 26, and then it just falls off a cliff. At 30 degrees C, we see larvae, and larvae are developing, but none of them make it out of the larval stage to adulthood. They die before emergence. So we can play a little bit with the tissues of those animals at 30, like at the thermal, like that achronic thermal limit. But mostly we're sort of interested in what's happening between this like 22 and 28 degrees C range, where everything just seems to fall apart. Interest in thermal tolerance and in a lot of the leading paradigms for understanding it is described by this oxygen capacity limited thermal tolerance, or this OCLTT. And when I first started reading this literature, I was really enthusiastic because it made a lot of sense to me. And my thought was, if we could, because we don't really know a lot about the thermal limits of most of our, particularly any of our species. And I thought, well, if all we have to do is just find the tipping point when animals go hypoxic and measure those metabolites, and we could just very quickly grind out thermal limits to lots of different species. The problem was that when we did those early experiments, we didn't kill the animals at environmentally relevant temperatures. They can handle short-term acute thermal stress at temperatures that are beyond ecological relevance. So, so we started to explore this a little bit further. So just in a nutshell, the idea behind this oxygen limitation idea is that as you approach the thermal limits of organisms, the aerobic scope, which is the difference between the maximum metabolic rate and the standard metabolic rate, and I show that on the panel on the right, shrinks. And so at those thermal limits, we get to a situation where oxygen becomes limiting. And in a little bit of work in aquatic insects has supported this hypothesis, but it's been done largely with experiments that have done that Deathramp related experiment, very rapid temperature increases at very high temperatures. And so we are interested in exploring if this oxygen limitation idea is actually functioning at the chronic thermal limits, because these temperatures are actually relevant. We experience them in our streams here. And so we started this with some respirometry. We had this like miniaturized fiber optic system. And this is basically what respiration rates look like for these animals on the Deathramp. So out here at 40, these animals have died. Each one of these colors represents a single larva, and we're serially measuring the oxygen consumption every few minutes. We're measuring the dissolved oxygen in the chambers every second, and we're taking the measurements every few minutes, and then we're moving on to the next measurement as we're raising temperature 1 degree C per hour. And when I look at those data, I know, and I see 30 degrees as the chronic thermal tolerance, nothing jumps out at me here as saying like, well, these animals are oxygen limited. They obviously have the ability to breathe more, but they're not. So that was one piece of evidence that that sort of led me to think maybe this oxygen limitation thing isn't really all that appropriate for chronic situations. And this evidence, when I first tried to publish it, was not well accepted by peer-reviewed people either. The other piece of evidence that we had at that time was we had sequenced a bunch of cDNA, and we found 2 genes that if we stripped the oxygen of water, the animals would express 2 genes for us. One of them is called EGL9, and this is a gene that controls HIF1 alpha signaling, that's hypoxia-inducible factor signaling, and the other is lactate dehydrogenase. And so these are sort of classic genes that are associated with hypoxia. We found HIF1 itself pretty constitutively expressed. It didn't really change very much even under hypoxia. We could get up to maybe a 2-fold change in expression, but most of the time it was even less than that. So our vision is that HIF is like constitutively expressed, and EGL9 is a much better marker for us. Now, if we look at expression of animals, now we're putting them on a thermal ramp again. This isn't a Deathramp. We're only ramping them up to 34 C, but again, it's like 1 degree C per hour. And the 22 C group is our control group, so all the gene expression is normalized to them. And as we warm, what you see is an increase in that green bar, and that's heat shock protein 90. So the animal's telling us we're feeling the heat and it's hot. And we also see that with heat shock protein 40, but to a lesser degree. But if we look at EGL9, it does nothing. It stays flat across the whole range. And we do get this one little blip of lactate dehydrogenase up at 34. This is past the chronic thermal limit. And it's not all that impressive in terms of the full change of its expression. The 22C with the number sign, that is basically animals that were held through this whole time course and then sampled again at the end, so that there wasn't any sort of issues about time of day or any sort of like clock related patterns. So when we try to submit this work, we found that it had a hard time being accepted, largely because we did not show aspects of aerobic scope. And aerobic scope is typically done by exercising the animals or putting them into a swim tunnel or something like that, with things that we didn't have. How do we exercise a mayfly in a respirometry chamber? We did not have a clever way to do that. So what we ended up doing instead is we used this agent FCCP, which poisons the mitochondria, and it results in massive, basically maximum oxygen consumption. And so the way we do these experiments is we've got the animals in these respirometry chambers, we get the background, we add the larvae, we measure standard metabolic rates, several of them over a period of time, and then we introduce the chemical treatment to the animal in the chamber, its oxygen consumption goes crazy and we get an estimate of maximum oxygen consumption. When we look at real data, it looks like this. So we have the standard metabolic rate shown in that lower dashed line with the open circles are animals that did not get treated. The closed circles are animals that did get treated. The arrow here in the middle shows when we introduce the chemical, and then we get this explosion of respiration. And so we now have a way to estimate maximum metabolic rate, and that difference between the maximum metabolic rate and the standard metabolic rate is the aerobic scope. And if we look at what we got for aerobic scopes across these temperatures, we see really no difference in aerobic scope, even at temperatures that are the chronic thermal limit of this animal. So at 30 degrees C, no larvae make it out of their rearing jars. But at that temperature, at least in the short-term exposure, there's no issue with oxygen limitation. So we did finally publish this work, but it was not without a battle and making us think about how to make the scope measurement. So our data does not support the OCLTT for chronic thermal exposures. If we go back, does anyone have any questions about that before I move on? Okay. So an alternative sort of perspective on thermal limits, or at least on how organisms perform over a range of temperatures, was articulated in this really important paper from back in 1980. When I was a student taking stream ecology, I read this paper. This is Bern Sweeney's work when he was a PhD student, and he was a student of Robin van Oot, and you would probably know his name as the person who invented the idea of the river continuum concept. So very heavy hitter in stream ecology. They articulated this view of development and development time and fecundity and fitness in terms of like a resource allocation perspective. And so in the middle, where that optimum temperature is, what they propose is this assimilated energy from the diet is broken up into respiration, which is sort of the cost of staying alive, I would say, and then growth, which is further differentiated into adult and egg tissue. Now, when things are hot, if we go down from that middle one to the non-optimum austral edge, or what we see is that the generation time shrinks considerably. That respiration piece on the bottom of that fork is a much fatter slice of the animal's assimilated energy, and less is there available to grow. What they proposed for the cold part was something sort of intermediate, and this caused some consternation to folks like David Atkinson, who were proposing the temperature size rule, and this didn't quite fit the temperature size rule at the colder temperatures, and there was a lot of attention spent on like, why is these mayflies different? It turns out that they're not. This is what the data they had at the time. These experiments have been redone, and it actually looks like this, and it fits the temperature size rule really beautifully. So the questions that we have is like, how does this work, and what sets these chronic limits? Like, still, what is the biology happening at that hot austral edge, or that non-optimal hot? Like, what makes the animals behave, perform so poorly? So this paper came out. This is a work from Burns Lab. So I was when I eventually got my professor job and started thinking about temperature. I started communicating with Bern, and it was just such a pleasure to, he was like, that paper from when I was a student was so influential to me. And so to be working with him on a project, kind of made me feel like I had arrived, as a scientist. But this paper came out just pretty recent, 2018, I think. And it's again based on a lot of rearing work. And these are basically the same species of animal, just raised at different temperatures. And you can see what a profound effect temperature has on body size. The big one was grown under cool conditions, while the smaller one was grown under hot conditions. And so here's some of the life history data that David Funk was able to generate, which is remarkable. So if you look at survivorship, it gets that sort of classic shaped curve. A very important shape here is the one in plot B, where it shows the development time, right? So under cold conditions, development time is really, really long. And if you drop down to the C panel below that, the mean adult dry mass is quite large. And that mean adult dry mass is just a straight regression to get us to panel E over here, is basically completely linked to fecundity. So bigger animals have more eggs, and it's really, really difficult to move animals off of that relationship. If we look at panel D, we see that the animals are growing. The growth rates are actually highest right before they fall off a cliff. And then finally, if we look at panel F, what we see is that across this range of 18 to 30 degrees in this animal, they're more or less following a degree day model perfectly. Like a certain number of degree hours or degree days gets you development, and it's predictable. When you get outside of that, though, those degree days expand actually, and when you get too cold, they actually decrease. What's interesting is that here's the per capita rate of population increase, which is very different from the individual fecundity, right? If we go back and look at the individual fecundities in panel E here, these animals that were, slow and fat and fecund, where individual reproductive output was maximized, it didn't really translate to population growth rates. They actually peak, really close to the thermal limits, which is really intriguing. We did a bunch of the respirometry work to support this paper, and we made lots and lots of measurements of insects breathing in chambers. Panel A shows the effect of both body size and temperature on respiration rates. Sorry that the axis is cut off on B here, but this is the metabolic rate in terms of micrograms per gram of body weight per hour. And then this panel C is Deathramp respirometry in some of these animals. And you can see the sharp uptick in respiration rates, out of 30 to around 32 degrees, which is sort of intriguing because around 32 degrees is about their thermal limits for this species, right? So that is like the tipping point respiration-wise of where performance really starts to fall off. So we were able to generate this sort of schematic cartoon of what we think is going on here. And Byrne coined this term acclimation zone, which is basically the temperatures where the animals are all conforming to the degree day model. So regardless of what the temperature is in this zone, their development is completely predictable based on just multiplying temperature by days and getting a certain number of temperature days, and that. You see the huge difference between the individual fitness maximization, that RO max at 16.2 degrees, and you see the population growth rate estimate max at 27.8. Was there a question?
You need to wear your good.
I did not hear that.
I think it's somebody who's just forgot to mute their microphone. So if you haven't muted your microphone, can you please mute your microphone?
Yeah, it's okay. I already muted them all. That's fine. Sorry.
Okay. Does anyone have any questions on this? So a tremendous amount of work by David Funk in particular went into sort of creating this this perspective. Now keep in mind that this is an animal that develops in summer and has multiple generations per year, which is why that R max value is so high, right? They're compensating for pretty poor individual fitness by actually squeezing multiple generations in. And if you're an animal that is one generation per year, you're going to suffer a whole lot more. So keep that in mind. So armed with some of these ideas about how the life history of the mayflies and development, play out, we wanted to bring this back to our animal, Neocloeon triangulifer. The data I just showed you was Cloeon dipterum. So this is a related but different animal. And so so we reared animals. This is again, Dave Funk led the work on this. Reared animals for their full life cycle. This is the data I already showed you previously for survival. Here's what the adult dry mass looked like. And again, it's sort of exactly what we saw with the other species. We maximize body size and fecundity at the cooler temperatures here. And then they are linear until they fall off a cliff. And then here's the mean instantaneous growth rates. And so you see those growth rates go up until they peter out. And so for us, we were like, well, we really want to know like what's going on in the tissues of these animals under these different thermal regimes. So again, we go back to that same suite of genes that we looked at before. This time though, they were sampled late in life. They had been grown up chronically under these different static conditions for their whole lifespan, and right before they emerge as adults, before they get the dark wing pads, they were sampled, flash frozen in liquid nitrogen, and we did this gene expression work. And so we see again the heat shock protein 90 pops up the way that it did previously, although much more muted in terms of like the magnitude of the increase. Again, lactate dehydrogenase and EGL9, those blue ones, they are hallmarks for hypoxia. And we do see a little bump up in LDH, but not statistically significant. So we don't really see systemic hypoxia signaling in a big way chronically here. What we do see though, which is kind of interesting, is this reduction in trehalose synthase. This is a storage sugar. And what we also see is this increase in insulin receptor, which is kind of regulating growth and development, and another gene called mTOR, which is very important. So we basically get the stimulation of growth rates and development at the expense of energy storage. We also did this targeted metabolomic approach here, and I'm just going to summarize this real quickly. What we found in the 28 group, this is like the edge of being able to make it to adulthood, is we see this big increase in biogenic amines. Histamine goes up, as does dopamine and dopa. All these, and at 30 degrees, these things are all increasing. We see a pretty good reduction in the quantities of certain lipids. So at 28 degrees, 8 lipids we found all decreased, and at 30, at that thermal limit, 12 of them, most of them decreased significantly. We see carnitine, 1 identified at 28, 6 identified at that thermal limit. So we're sort of getting at what's different between making it and surviving under thermal stress versus not making it and surviving under thermal stress. And these are the things that popped out at us. We also see this amino acid, these amino acid changes, and which we'll talk about briefly. So how do I get rid of my bottom taskbar here?
I'm not sure, David, but the bottom is, you can't see the bottom in my screen. I'm not sure how that.
Yeah, let me see if I can get rid of that. Anyone know?
Potentially move your or switch your screen from the Your video being on the top to the side? Unless that's
Could you have the screen which is showing not take the entire page up, entire screen up? Would that help? I'm not sure if that would.
I don't know if I know how to do that. I think I'd like to just be able to turn this thing off. How do I do that?
I really don't know. I've never seen this problem before, so.
I had it off. Anyway, well, I apologize for that. I'd like to be able to turn that off. I'll just move on. So one of the things that we saw that's pretty interesting is this ratio between glutamine and glutamate. And at 30 degrees, at that thermal limit, we see that they're twofold higher than those ratios, more than twofold higher than those ratios relative to controls. And what Tina thinks is happening here is that in the absence of glucose, in the presence of oxygen, cells could switch to something called glutaminolysis, where the TCA cycle is being filled with glutamate. And so basically you have aerobic glycolysis, you basically switch to this alternative fuel supply, which is sort of an interesting finding. We also find a decrease in available sugars, right? So, so the animal is like looking for something else to burn, and these are some of the things that we found. In terms of potential biomarkers of thermal stress, we see histamine as a pretty good biomarker that moves in relation to temperature and is linked to predicted fecundities based on the sizes of the animals at emergence. And so we don't really have a predicted fecundity for this one, because that's why it sits on 0, because there is no fecundity at 30 degrees. The other thing that we see is a temperature sort of specific, a nice temperature response with this lyso-PC lipid. So yeah, so basically these results show us that the genes related to energy metabolism suggest the larvae were energetically challenged. We see a general reduction in the lipids and acyl carnitines, suggesting utilization of alternative energy sources. The gene expression and the metabolomics don't really show evidence of hypoxia at those thermal limits, and we've identified 2 potential biomarkers for thermal effect studies. So we've now done 2 scenarios, right? We've done like the Deathramp, which is if you want to get it to where the animals actually die, you move into temperatures that are not environmentally relevant. And then we've done the difficult task of the chronic rearing across all these temperatures, but that's also not very realistic either, because animals aren't experiencing constant temperatures in the world. So unless you're living in a spring creek somewhere where the temperatures are pretty stable, you're going to have seasonal variation, you're going to have die yield variation. So the next step for us was to move towards this die yield thermal change. And we know something like during the day, it's going to heat up, and during the night, it's going to cool down. And we were sort of interested in asking the question is, how do life history outcomes vary with die yield temperatures relative to static temperatures? And how does gene expression change within and across some of these thermal regimes? And we had the question of, does more time spent at favorable temperatures compensate for time spent at stressful temperatures, right? So if you have more of the good temperature, but you also get a little bit more of the bad temperature, like what wins? And so here are the full set of exposures for the rearing experiment. And each of these exposures varies by 5 degrees C per day. And they hit this maximum temperature and they sit there for 2 hours and then they come back down again. And what's nice about this regime is that the average daily temperatures can map directly back on top of those static temperatures that we did. So we can compare them directly. Again, this rearing work was done by the Stroud Center, and they are working on a paper to show in more detail these life history outcomes, but I can share a few of those results with you here. So just to remind you, these are what the static rearing study showed. And this is the same data I showed you before, just in a bar graph form. Again, survivorship falling off a cliff between 26 and 28, development time dropping considerably, and then increasing again under serious thermal stress, and the very significant effect on adult body size with temperature. And this is what we see with the diel results. Same general patterns, but notice in the development time plot, for example, how much at 28, how much that diel regime sort of influences that. And we see this similar pattern with body size. So in comparison, and comparing them directly to each other now, these bottom set of plots show the percent difference between the diel and the static. So basically the diel animals were not significantly different in survivorship at the 2 cooler temperatures, but at 28, the diel situation was much worse in terms of survival. Development time difference did not change in the sort of heart of where they like to be at around 22. But relative to static, there was an increase in development time in the 26 group. So experiencing stressful temperatures delays development more. And that is even more pronounced at 28 degrees. And then finally, we get a really pronounced effect on final body size. More time spent at high temperatures definitely is not offset by more time spent at favorable temperatures. And you see those size deficits. And so I think this is a really cool data set. I'm not aware of any other ones that have this direct comparison in aquatic insects at all. What we did on our end is we focused on these 2 groups, right? We knew that 28 degrees was stressful in that chronic situation. And I was really curious about what happens when the animal on average has a pretty good temperature, but experiences these stressful temperatures transiently. And so I'm comparing basically 2 groups here. And what we did is basically we took an RNA-seq approach. And so these are the 2 thermal regimes. The red one represents the 26 degree mean temperature, and the blue one represents the 22 degree C mean temperature. And basically the larvae were raised from egg hatch to late larval maturity. And at that time, we pulled samples for gene expression. So those letters on this plot represent the sampling points for gene expression. And the blue one represents diel variation within what Byrne calls that thermal acclimation zone. So the animals are wiggling between two nice temperatures for development, whereas the 28.5 is outside of that thermal acclimation zone. And so we know that it's stressful, but we're only giving that stress transiently. But they're spending a lot of time the rest of their day, in pretty good temperatures. 23.5 is good. So the methods, real briefly, the key thing here was to sample larvae at comparable developmental stages. And so that was sort of a tricky thing to do. We did this kind of visually by looking at the state of the wing pads. So they were still kind of light. They hadn't turned dark yet, but they had a certain level of development. And we said, okay, they're close. We then sequenced 16 libraries. So it was basically 4 libraries per, basically 4 replicates per temperature treatment. Were sequenced with paired end reads using the Illumina NextSeq 500. We constructed a de novo assembly, transcriptome assembly using Trinity, BLAST Plus for annotation of the genes, and then we try to do reactome pathways and some pathway analysis. And then we did some reality check comparisons with qPCR to make sure that the gene expression data that we were seeing in RNA-seq was matching what we would find with qPCR as a QA check. And so here is that QA check. I don't remember off the top of my head what Tina's rationale was for choosing these genes, but the black bars represent the RNA-seq data, the red bars represent the qPCR, and when we plot them, there's a very nice correlation between the two, so we feel pretty good about the sequencing bit. And here we have 3 comparisons I'm going to show you. That we'll start with A and B. And so this is thermal variation within the acclimation zone, and we see very few genes differentially expressed, either up or down regulated. So when the animals are wiggling between happy temperatures, very little is going on, and those genes that are being differentially expressed, a lot of them have to do with day and night, vision-related things, and there were very few of them. The other initial comparison we wanted to make was C and D up here, and we have a boatload of differentially expressed genes, both up and down regulated. And then finally, the third comparison we made is this one in the middle here between C and B, which is basically animals were sampled at comparable temperatures, but they belong to very different thermal regimes. And you can see that in purple here, that the residual, we sort of think about this as the residual effect of daily forays out of the acclimation zone. So what sort of signature of this transient thermal stress are the animals carrying with them, even when they're at a comfortable temperature? And so basically the top bubble plot here is a list of the processes that we went, using different Go categories to sort of group genes by function. And the blue circles are downregulated. The red circles are upregulated, and the size of the circle sort of speaks to the magnitude of genes involved in that process. And I'll talk about these results in a little bit more easy way to see. So during that thermal push, so basically we're talking about this this top bit. What we see is the upregulated process of lipid and carbohydrate metabolism. This is exactly what we see in the chronic rearing studies when we did the metabolomic work. We see the lipid, and so that makes sense, right? The cost of staying alive when it's hot is higher. And we see a whole lot with chitin metabolism and chitin development, basically the stimulation of development and molting. We see increase in sterol transport and in deactivation of rhodopsin-mediated signaling. And then in terms of the major downregulated processes, we see N-glycan processing and some immune-related stuff. So I'm going to do a little digression here. I was really curious about this chitin stuff because we've noticed that that when we rear animals in well plates sometimes, that their molting does not seem to be dependent on size. So we did these experiments where we raised animals in individual well plates and we counted their molts every day. We were looking for molts, and we did this at 2 different temperatures. These are static temperatures. But if we look at this plot over here with the average number of molts, there is no difference in the total number of molts that these animals made over development. But what is interesting about these 2 different temperatures is that, at 18 degrees C, those animals are big and they took a long time to develop. They took 40-something days to develop. So whereas at 26, those animals were small. They only took 20-something days to develop. And so if you look at like how many days between molts, you know, at that, you know, these animals at 26 are molting more frequently. But totally intriguing that the molting isn't stimulated by size. So these hot animals are molting at smaller body sizes and more frequently. And so if you think about like the caloric loss of taking all of that tissue and shedding it, but it's not driven by growth is really fascinating to me. And I'd love to understand why the seemingly wasteful biology is happening, but I don't have an answer for it. So we see an relatively equal number of molts, but we don't know why the hot larvae molt at such small sizes. Like, what is the advantage? Speculated that maybe there's some possibility that that there's some excretory function associated with molting that we don't know about, or possibly that it's just neuroendocrine determination of molting that is just, it happens whether it's advantageous to the animal or not. I'm not really sure. We tried to collect exuvia for a comparison of the energetic loss in terms of like calorimetry, but we just did not get enough material to do that. These animals are really small. Anyway, going back to this other regime, this one on the bottom now. Now here we're comparing this guy in the middle, right? We're sort of looking at is this residual effect of this chronic daily thermal challenge. And what we see is that the animals from C, from that 26-degree mean group, they have reduced ATP synthesis. They have, what's interesting there is they have increased lipid synthesis. So remember, at point D here, the lipids are burning up and they're using these lipids really rapidly. So at 23, they're like working really hard to put that lipid back on, which is really fascinating. We see increased synaptic vesicle transport and neurotransmitter secretion. This is, again, we saw that in the metabolomic stuff with the histamines and the other biogenic amines. And so that was interesting to see that again with a completely different way of exploring it. And we also see an increased regulation of membrane potential in these thermally stressed animals. There is still really a lot to learn about how all this stuff works, but I think that this energetics perspective that Van Oot and Sweeney articulated in 1980 is generally supported by some of these sort of modern biology approaches. And I think that it probably does have a lot to do with energetics more so than oxygen related issues. Let's see. I just have a few minutes left here. So we have obviously we have issues with temperature that are not going away anytime soon. And with the fires burning in California, we had Trump telling the governor of California, don't worry, it will get cooler. So I'm assured. Anyway, there's going to be a lot to learn about temperature directly affecting organisms and temperature interacting with other stressors. So real quickly, I think that temperature toxicant interactions are super important and underappreciated. I have a student in the lab now, Sarah Orr, who's doing fantastic work. She just published this year this paper about temperature influences in ion transport and toxicity. And I'll just fly by this really quickly. So basically what we do is we do these experiments with isotopes. So this would be sodium-22. We put these animals in exposures, pull them out, rinse them, count them on the gamma counter, and return them to their exposures. And we get rates of increase in sodium uptake over time. We take those slopes, and those slopes are what I'm going to show you in this next slide, where we look at sulfate sodium and calcium is the far right. That plot that I showed you just now is this particular data point. This arrow is the previous slide. We see a few things here, some species-specific differences in just transport rates. We see across the board that calcium transport in these aquatic insects, the rates, if you look at the y-axis, the rates of this are very, very slow. And that is kind of uniform across the board. Like all these animals move very little calcium. Sodium is huge though, right? There's a ton of sodium movement in all of these animals. And what I have on this x-axis, if you can see it, are temperatures. So 15, 20, and 25, and you can see that temperature is massively increasing the flux rates of these major ions, and that the major ion transport rates are scaling with temperature. So the first plot, I've got the standard metabolic rates at these temperatures. The second one, I've got the sulfate uptake rates at these mapped to the standard metabolic rate, and then I've got the sodium uptake rates. And you can see that metabolic rate is sitting right on, like the relationship is really, really nice. So we know what metabolic rate, how it responds to temperature. And so clearly the animals are taking up more of the major ions at higher temperatures. And this does translate loosely to toxicity. Here we have percent survival in these animals, and it's not perfect. The 15-degree temp guy is intermediate here, where we might expect it to be flat. But 15 is also potentially a little bit cool for them. So it's possible that that is a modifier, but certainly between 20 degrees, which is a very nice temperature for them to develop in, versus 25, a huge difference in survivorship. And so we see that temperature modulates the toxicity of sulfate, and I suspect it's going to modulate the toxicity of any other kind of major ion matrix. So we know that water temperature increases metabolism, ion transport, energy consumption, major ion toxicity. We think that the energetic cost of osmoregulation in warm, saltier environments may cause significant major ion toxicity problems in some of these animals. We've also done a little bit of work looking at the interaction of temperature with some insecticides. These are some papers that we've put out in relation to the pesticide imidacloprid. In both cases, increasing temperature increases the influx rate of the contaminant and increases the toxicity of the material. So again, we have a lot to learn. And remarkably few scientists are doing controlled experiments with aquatic insects, despite their critical importance for freshwater ecosystems. We have a lot of field observational things going on, and not a lot of folks trying to drill down into mechanisms. So that's one of the reasons Ben and I are always in touch with each other, because we think about some of the same problems, and we have a lot of shared interest in this territory. So with that, I want to say thank you for your attention, and recognize some of the students and folks at NC State who helped make all this go. Dave Funk, who's my best collaborator at the Stroud Water Research Center. Some folks at Research Triangle International who helped with the metabolomics. And with that, NSF for funding, and with that, I'd be happy to take any questions.
Thanks, David. That was fantastic. That was really great, really interesting.
I know I kind of moved through things, and I apologize about the not being able to see the bottom of the slide, but
no problems. Who's got some questions? Either type them into the chat or turn your microphone on and ask the question that way, whichever you prefer. B.
Hi. Yeah, that was great. Thank you very much. I have a question about your dial variability experiments. As I understand, you had mean temperatures between 14 and 28 degrees and had a temperature variability of 5 degrees. And I was wondering how you choose those different temperature ranges and how long you run each of those experiments. And if those temperature ranges were based on stream temperatures you measured beforehand, or if that was just based on literature you found.
No, so those temperatures were chosen. We wanted diel variation, and we wanted it to be environmentally relevant and meaningful. And the degree to which water temperatures change on a daily basis can, it scales to the size of the stream. Typically you see, 5 to 8 degrees C change per day in many of our temperate streams here in North America. So we took a fairly conservative look. We wanted it to be a relatively modest amount of change. We also wanted the average temperatures to map back to what we had done on the static exposures. So we could lay them right on top of each other and say, like, how does the way that the animal experiences temperature, affect its, development in biology. Yeah, that was the rationale there. How do I get to seeing you all?
If you press there's a single, there's like an array of 3. That's it. Okay.
Okay, there I got you. Okay.
So there's a written question, but just before I'll read that out. But just before I do, I had a sort of related question, and that's with the 28 degrees daily variability. It's fluctuating up and down, and you're interpreting that as they're having rests in the more comfortable zone in the lower temperature, but they're also having excursions into the 30 degrees, which are lethally or long-term lethally top. So why do you interpret that as having a rest in a lower temperature? You could equally interpret that as having excursions into a problematic temperature.
Yeah, so that top one we didn't look at. The larvae did not do well, right? They were pretty beat up. So we focused on those two. There was another temperature treatment, which is the one you referred to, above that, which we didn't have the samples to play with very much.
Okay. There's a question it says it's from Kyle. Why don't insects produce antioxidants in response to heat stress?
I thought that they would. In fact, we developed PCR primers for the usual suspects of antioxidant enzymes and found that they didn't really do much for us. And so I fully expected more oxidative stress under high temperatures. And was surprised to at least in our hands, we did not find evidence for that. Theoretically, it makes sense. Like, you should see it. So I don't know. That's good. Great question.
Okay, thanks, Kyle. Any other questions? Either written or turn your microphone on. Yep.
I have a question, if I can.
Of course.
Yes.
Oh, excellent. Yeah. Thank you. I was very interested on the final section of your talk where you talk about ion fluxes and changes in the rate that the insects are taking in and I guess exposing those ions. It makes me remember a talk that we saw earlier in the year by Dr. Bob Wong from Monash University, and his area of expertise is on, I guess, ecological impacts of pharmaceutical components and pharmaceutical pollution in the wild. Maybe really, it made me think, have you considered the possibility of investigating how these pharmaceutical pollutants in the environment could be affecting microinvertebrates such as these species with increasing temperatures or, I guess, fluctuating temperature environments?
Yeah, I mean, there'd be a lot to do on that front. I know Emma Rosie Marshall has been thinking about this problem a lot and doing a lot of work on in urban streams, looking at pharmaceuticals and such. Yeah, it's certainly an area of that would make a lot of sense, especially in urban environments. You know, frankly, my research program shifted a lot from like straight contaminant stuff to more of this physiology stuff because I was having a really difficult time finding funding for the contaminants work, whereas NSF was a place I could go for you know, physiology. And so I really just kind of put my eggs in that basket more so just to stay alive as a scientist, honestly. The opportunities in the United States to work on contaminant related problems are really limited. You either have to cozy up to industry and make relationships there and do some of their bidding, or you know, the EPA has vanished in terms of supporting extramural research on contaminants. So there is no place to go for support for that right now.
Okay, thank you.
Any other questions? I'll ask one in terms of the molting. David, do you think that maybe that they've just got a certain number of molts that they've got to get through to complete their life cycle? And maybe that's based on, you know, oxygen throughput through them or something like that? Because, I mean, I think some caddisflies just have to, they all have the same number of molts to get through their life cycle, and maybe mayflies has got something similar, and there's sort of total pre-metabolism which they've done is sets the trigger for the for that molting.
Yeah, I just don't, you know, if your goal is to grow big and produce as many eggs as you can, why are you throwing away all these assimilated calories if you're not big and you don't need to molt, you know? Like that's the thing that I struggle with. I just don't understand, like, it just seems so inefficient, you know?
But if they haven't been exposed to these temperature stresses in the past, they may never have evolved that capacity because they never needed to.
I don't know. That's a good question.
So I do realize we're over time. So if people need to leave or want to leave, then please feel free to. If there's any last minute questions for David, then please ask. Again, either by typing or coming on screen.
Again, if anyone thinks of anything later on and wants to send me an email, I'd be happy to correspond with you. So this work with the RNA-seq and the diel fluctuations, that paper is in revision right now. Actually, the revision was just submitted a couple days ago. So hopefully they'll like it this time. And we'll have a paper to show for the work. But I'll let Ben know when that happens.
We'll look forward to seeing it in its final form. It'd be great. Okay, it looks like there's no more questions, but please come on if you want, and we'll all thank David. It's been a great presentation. Thank you so much.
Thank you all very much. Appreciate the opportunity.
Cool. Okay, well, we'll see you all later. Hopefully I'll see you. And before 4 years, the situation in the world will allow travel, and we'll better see each other face to face sometime between now and 4 years.
It's been a crazy year.
Yeah, sure has.
Well, thank you very much, Ben, for the invitation to speak. I appreciate the opportunity.
No problems. We appreciate it.
We will be uploading the video today to our science communicator officer, and after that, I believe she will be uploading that to YouTube. So we'll be sure to share with you the link. Thank you so much for your time. I do appreciate that it's quite late now where you are. All the best and stay safe.
All right. Take care. Thanks, everyone.
Thanks,
Thank you,
David.
David.
Bye.
Bye.